Hi, my name is Nandita Jayaraj and today I have here with me Sravanti Uppaluri. Hi Sravanti.
Hi.
So as you all know, earlier this month was Nobel Week, and for us here at Azim Premji University, that's an opportunity to do some cool science communication. Sravanti, I've invited you here to talk about the 2024 Nobel Prize in Physiology or Medicine. This year, the prize went to Gary Ruvkun and Victor Ambros for their discovery of something called microRNA and the role of microRNA in post-transcriptional gene regulation. So we're going to get to know all about what this means through the next few questions. So I think it's safe to say that almost everyone these days has heard of DNA and most of us have even heard of RNA, but microRNA is a little bit of a foreign concept to us. So can you break that down for us?
MicroRNAs were interesting because I think when we initially discovered, the structure of DNA, when Watson and Crick discovered the structure of DNA rather, I don't think we ever had in our imagination, right at the beginning, what something like microRNA could be. It turns out that, what we typically know about DNA, and as you said, everybody's heard about DNA. So DNA is a set of instructions, and all of our cells in our body have the same set of instructions, right? But the thing is that even though, let's say, my skin cells have the same set of instructions as the cells in my liver, let's say, right? The cells are completely different. They perform completely different functions. So how does that happen? And that happens essentially through a process called gene regulation, right? So that's one part of, the title of this thing, the prize. So how do genes get regulated, and what does it mean for genes to get regulated? What that really means is your DNA is a set of instructions, a set of genes, basically, right? And genes are things that code for proteins. Okay, and the way that it codes for proteins is that DNA first gets transcribed. So that means it's almost like a copy is made, a subset of the DNA, a copy is made into RNA. Okay, and that RNA then gets translated into protein. So it's a kind of a two-step process, right? You can think of it. DNA gets transcribed to RNA, RNA gets translated to protein, and those words really are chosen well, right? Because it's really what we think of when you're doing a transcription. You're copying something down. When you are translating something, you're changing it from one language to another, right? So in the case of RNA to protein, you're changing it from being a nucleic acid, which is what RNA is, and then you're changing it to, you're sort of decoding it, you're translating it into the language of proteins. So what microRNA is, it's helping to regulate this process where you're going from DNA to protein, right? So if you think about it this way, I have a set of instructions, and my skin cells that I alluded to earlier follow some subset of those instructions. My liver cells may follow another subset of those instructions, and that's why they're different from each other, right? But what is it that regulates this difference in which set of instructions should be followed and when, right? That's a huge part of how genes are expressed and regulated. And so this process of going from DNA to protein, that's where microRNAs come in, right? MicroRNAs are basically themselves RNA that are transcribed from DNA, and they're kind of floating around in the nucleus of a cell. And what they do is that they are complementary to some RNA, other RNAs.
Okay.
So these RNA molecules normally code for proteins, so they would get transcribed and then translated into protein. But if you block that step, right? If you say, I'm not going to let this RNA get translated into protein, you are blocking the expression of a particular gene. Mm-hmm. And so these microRNAs are complementary. In other words, they can go and bind to these gene expressing or gene coding RNAs. They can bind to them and prevent them from getting translated. Right. So that's what a microRNA really is. Okay. And it's called micro because they're very short. They're just 22 nucleotides in length. And compare that to, let's say, a gene that might be thousands of nucleotides in length.
I see. So it would be correct to say that a microRNA plays a role in the decision that a cell takes to become a skin cell or a heart cell.
Yes, more or less, yeah. So it's actually the discovery of microRNAs was actually made in the process of trying to understand aspects of developmental biology, which really is referring to how cells make decisions, right? What kind of cell should they be, and how do all of those decisions come together to form, let's say, a functional organism, right? And so that was really the process through which microRNAs were discovered. Later on, of course, there were many other discoveries where they were found to be important.
Can you briefly tell me about Victor Ambros and Gary Ruvkun's role in getting to know all of this?
Yes. So I'm not really an expert in their history per se, but they both actually were working in Robert Horvitz's lab. Robert Horvitz himself is a Nobel Prize winner, also worked with the same model organism, and he studied different aspects of, again, developmental biology, and also studied this process of cell death called apoptosis. And Dr. Ambros was working on looking at what, let's say, gatekeeping aspects of developmental biology. So he was using a model organism called Caenorhabditis elegans, also called C.elegans. And he was trying to study how this worm develops through its different larval stages. And so he was able to identify a particular gene where if it was knocked out, called Lin4, or rather he was able to identify that when Lin4 was knocked out, development didn't proceed properly, right? And Victor Ambros and Gary Ruvkun were postdocs together in the same lab. And together they actually were able to identify some of the key players in the process of this, say, larval progression in worms. Okay, that by itself, said like that, doesn't sound so great. But what is really interesting is that they were able to first identify that Lin4, which, so when they worked together as postdocs, they were able to actually show that when this particular when Lin4 is knocked out, larval development doesn't proceed properly. And then they also showed that another protein, when it was itself was also knocked down, then they could compensate for the first gene being knocked out, right? So there was kind of this domino effect. Later on, what Victor Ambros showed was that Lin4, which was the original protein, or the original, say, candidate, I should not say protein, what they found out was that Lin4 was never actually translated into a protein. They saw that it was just an RNA molecule, and it being RNA was enough. So knocking out that the ability of the cell to produce this RNA was enough to block larval development. So it never got translated into a protein. So this basically indicated that it wasn't just proteins that regulated processes like, gene regulation and cell differentiation, development, and so on, but also an RNA molecule could control the fate of a cell. And then basically Gary Ruvkun went on to, along with Victor Ambros, found out what the target of this Lin4 RNA molecule was. So what did it bind to? How did it work? And so they were able to figure out the mechanism, and they called it a microRNA because it was very short, just 22 nucleotides in length. What made this discovery all the more interesting was that they found that there was yet another microRNA in the same pathway, in this pathway of larval progression in C.elegans worms, that had a similar sort of control. So when you go from the first larval stage to the next larval stage, there was one microRNA that helped the progression. And then in future larval development, so say L4, the fourth larval stage to the adult stage, they were able to identify yet another microRNA that was essential for larval progression. And once they published this series of papers, this was over the course of 7 or 8 years, what they then did was, and this was again, these discoveries in the late 1990s, early 2000s coincided then with the genomics revolution. So now, all kinds of genomes were being sequenced, including the human genome. And so what they were able to do was quickly say, okay, I have this sequence of the RNA. Does it also exist in other organisms, right? And they were able to identify that, yes, it exists not only in humans, but, in snakes, in in scorpions, in mice, whatever, all these kinds of different organisms, they also exist.
If not for this, it would just be something really cool happening in worms. But now we know
Exactly. Right.
It's
so that's
in every
it is
organism?
in most multicellular organisms that I know of. Yeah. So it's really a very universal process, right? And so that's what made this discovery suddenly much, much bigger.
Right.
And so, in the 2000s, early 2000s, we went from knowing about 2 microRNAs in the worm to, over the course of like 2 or 3 years, knowing about thousands,
Wow.
right? That had really extremely varied functions, right? And this was also a very important discovery in terms of how we thought about genomes themselves. So as I said, in the early 2000s, where there was this revolution with genome sequencing, right? And it was very surprising when the human genome came out and they found that only 2% of the human genome had protein coding genes. The rest of the genome they thought was kind of junk, right? And then this discovery showed that, okay, the rest of the genome also coded for things that were important, even if they weren't for proteins, right? So microRNAs being part of a larger class of non-coding RNAs. Non-coding means that they never get translated into protein, but they have really essential biological roles. So that's really what they helped with. Yeah.
This particular prize seems to be one that deals with very fundamental biology. But are there also any potential applications that microRNA can lead us to?
Yeah, so the fact that we've been able to identify that microRNAs are really essential for many biological functions, right from things like cell division to the function of the beating of the heart and so on, means also that now we've identified targets, right? We know, where if things go wrong, we potentially know how to fix them. So there's lots of applications in human health and disease, definitely. And there are actually clinical trials going on with microRNAs.
Lastly, Sravanti, I know this particular Nobel Prize would have naturally excited you because you are a biologist, but there is also, I imagine, an extra thrill that you would have gotten because it's a prize given to a discovery that came from this worm called C.elegans that you described. And this is a worm that you also have worked with in your research career. So can you tell me a little bit about the worm? Because I heard that this is not the first time it's winning a Nobel.
It's the 4th time, yes. Okay, so one thing is that I think most biologists who work in the lab have this very strong affinity for their model organism, right? So I started working with C.elegans when in my postdoc, and before that I had worked on much smaller organisms, so things that were very, very microscopic, much smaller. And the worm is 1 millimeter in length, so it's also quite small. But compared to somebody who works with, let's say, bacteria, it's much bigger, right? And so I was first very excited that it was quite big. And so much of our identity as biologists, wet lab biologists, is actually tied with the model organism that we work with. So it's of course, it's very exciting. You develop this kind of affinity for them. And also it actually is a kind of a personal connection that you develop with your model organism. And, as you have students, they also develop an affinity towards the organism, and it's a kind of a community that spreads. And very often when you hear, actually, even if you hear Gary Ruvkun and Victor Ambros speaking about C.elegans, they are always talking about the C.elegans community, right? So it really is kind of whatever model organism you work on, it kind of is a community. So it's very exciting. What makes the worm an interesting system to work with, to give you a more practical answer, is that one is that it's completely transparent, right? So this means you can look at the organism and see what's happening. So it's not like us where you can't see through our processes, right? So it's transparent. So if you look through a microscope, you can actually see a lot. It has, so it's often referred to as the most simple complicated organism in the sense that it is multicellular. So it has specialized organs. So it has an intestine, it has gonadal cells, it reproduces sexually, it has a nervous system. So it has all of these things. You can study multicellularity. The other thing that's really interesting about worms is that it has what we call a very stereotyped lineage. I'll explain what that is, but this is especially important for developmental biology.
Okay.
So, a worm starts off as a single cell, just as most multicellular organisms do. And then it divides. So one cell goes to 2 cells, and then 2 cells go to 4 cells, and so on, right? But the cool thing about C.elegans is that, so the first cell division takes place, it's slightly asymmetric, so one cell is slightly larger than the other.
Okay.
And then I can predict, now that I know that there is an asymmetry, so someone has sat down and actually won a Nobel Prize for doing this, has figured out that as these cells divide, they have a very stereotyped cell division process or lineage. That means, let's say the cell on the left, I will know that this will be the precursor to the cells that will eventually become, say, the intestinal cells, right? And not only that, I also know that as the cells are dividing, I know that every single worm, I know where it will divide, I know when it will divide. Every single C.elegans worm on the planet, if it has developed properly, has 959 cells. So unlike you and me, we have very different cell numbers and we don't know exactly how many cells we have. For adults, for worm adults, that's the case, that you have 959. And this means then that if I can predict when things are going to happen, if something's gone wrong, I can then identify the genetic origin of that mistake in development, right? So it's a really cool system and tractable system to look at development. And of course, many of these genes have homologs in humans, so there's lots of things that we can tie together.
Thank you so much, Sravanti, for helping breaking this down for us.
My pleasure.
If you enjoyed this conversation I had with Sravanti, I have done a lot more of such explainers of science awards that have taken place over the past few years, and you can find them all on Azim Premji University's website or their YouTube page. Thank you for watching.